Hello everyone, my name is Abhishek and my teammates are Pranav and Tutesh. So this is our final year project and the topic of the project is brain tumor detection and comparison using CNN, SVM and XGBoost. So brain tumor is basically haphazard growth of cells inside our brain which can be caused due to various external factors and genetic factors which ultimately is really hazardous to our health and cause deaths and has given problems to mankind since ages. So what we are we are using CNN, SVM and XGBoost algorithm in order to classify and given MRI images as tumorous or non-tumorous. So firstly we are going to see basically how a CNN works. So basically a CNN has two stages. First is feature extraction and second is classification. In a feature extraction, what happens is that a set of pixels from an entire image is taken out, and from that a feature extraction is done. This is done several times through the entire image, creating a effective feature map which it uses to classify, in this case, a brain tumor or not. Then comes the classification layer, which has a fully connected layer which contains an activation function in order to classify whether the given data is correct or not. Yeah, we have used a ReLU activation function and the softmax function in our fully connected layer. Next algorithm is SVM or support vector machine. In this algorithm, what it basically does is that it plots various data points on a graph in order to effectively group the data. It creates an hyperplane beyond which it says whether an hyperplane below which it will say that it's not a tumorous, and if the data is above that, it will basically tell us that the image contains tumor. So what it does is that first we have a training phase, and the second we have a classification phase. So first what it does is that using the data which is pre-processed, and which basically means that the data is free from any data which we do not need. From this, segmentation is done and labeling is done. After that, it goes to the training algorithm, which feeds into the SVM model. After this, the model is trained. We get to the classification, whether where we input our data, which basically means input our image, which is to be shown whether it basically inputs the image which is to be checked has tumor or not, and using the pre-processed model, it classifies whether it is or not. Then what comes is XGBoost algorithm, which basically means extreme gradient algorithm. Unlike other boosting algorithms, means where weight of misclassified branches are increased, in gradient boost algorithm the loss function is optimized. XGBoost is an advanced implementation of gradient boosting along with some regularization factors. It falls under the category of boosting techniques in ensemble learning. Ensemble learning consists of a collection of predictors which are multiple models to provide better prediction accuracy. In boosting techniques, the errors made by previous model are tried to be corrected by succeeding models by adding some weights to the models. So this is what XGBoost is in short. Now we will come back to our algorithm. So this is a GUI which we have designed in order to interact with our code in the backend. So here we are going to select image using the select image button, from which we will take one of our images from our test set and see how, and then image pre-processing is done, and here it has extracted the part. Then we will do a scene and prediction. Here it tells what is the data that we have, it tells what level tumor that we have got is here. Then we will train our SVM model using the SVM model button, and we can see in the backend that here it is getting trained. It will take some time. And this is the final result from the SVM model that we have got, and this is how it is the image is plot upon the graph. Now we will train our XGBoost model. So here it shows that the SVM model has been done successfully. Now we will train our XGBoost model. It takes place the same way. It takes some time, and then it will show us our result. It does take some time to train the data, to train the model. And here is our final result for the SVM, for the XGBoost model. And here we can see that we have trained our model successfully. So this is how it goes in the front end. Now I will show you our backend and what code we have used. So the GUI which we are seeing was implemented using the tkinter function that we have in Python. So this is a GUI code. Which is not that important. So we will move on to our scene and model. In scene, and what we have is that we have used different libraries such as Keras, NumPy, and TensorFlow. These are predefined libraries in Python, which we have, which is used in machine learning. So we have used a sequential classifier also. Then comes our first convolution layer, in which we have taken 32 images at a time, and then an input shape is given, and an activation function known as a ReLU, which acts as a threshold for our convolution layer. Then we have used a max pooling 2D function for our pooling layer. This is done over 6 times in order to get the required accuracy. Then we come to the flattening layer, where the data is flattened using the flatten function. Then we again used a softmax activation function in order to get the data in 1 or 0 format. Then here we compile all the data. Then the complete image is fitted into the CNN model, where the data is made fit for the CNN model that we are going to use. So here as you can see, we have used 250 epochs, which means it will take it will do the same process 250 times for classifying an image. And the data which you can see here is implemented using these functions and using this code. So this is what basically CNN is implemented. Then we come to the SVM. In SVM, what we have done is that we have used pandas library, matplotlib library, in order to plot the graphs. So I have used sknode.metrics for the classification report and for getting the confusion matrix that we desire. In SVM, what happens is that some amount of data is used for training purposes, and some amount of data is used for testing purposes. So as you can see, we have different variables which stores the data for training and testing. For as we go further, you will see that some data has been stored for prediction purposes also. So this is where we have called our SVM model, and this is where we get our results. And at last we dump all the data in this joblib file. Now going forward to the XGBoost model. XGBoost is more or less similar to the SVM in terms of the image processing that it requires. So the implementation is more or less similar. It's just that we call on the XGB classifier in order to classify our images. And this is similar. This is also similar, where we have used different metrics function in order to get our numerical results and parameters. And this was our project, brain tumor detection comparison using CNN, SVM, and XGBoost. I would like to thank our guide, Mr. Vivek Ogle, and thank you everyone for watching this video.